J. Thelen, a Bond LSC researcher, next to the GCMS machine in his lab, which tracks seed oil measurements. Photo by Mary Jane Rogers. Bond LSC. How to get crops to produce seeds with more oil by M.J. Rogers. Bond LSC. You can get blood out of a stone, but J. Thelen wants to find ways to get more oil from seeds. We're specifically working on the metabolic engineering of oil seeds. Broadly, trying to increase the oil content of crops and raise the value of the seed in the process, said Thelen, a Bond Life Science Center researcher. Seed biology and metabolic engineering have long been interests for Thelen, and his lab combines biochemistry with cutting-edge proteomics technology to identify new regulatory modules for key metabolic enzymes. But let's start with why seed oil is important. Seed oil is big business, enough so that scientists are trying to maximize the amount of oil that seeds produce. It represents an important, renewable source of food and feedstocks, used for everything from salad dressing to combustible fuel. More than 448 million tons of oilseed crops were consumed in 20,152,016, according to USDA Economic Research Service. The oil essentially comes from lipids, or fats, in plant seeds or fruits. All plant cells contain lipids, and embryonic cells within young seeds are poised to make an abundance of them, especially the storage lipid triacylglycerol. While tree nuts can be up to 80% oil, most oilseed crops store 15 to 45% oil within the seed. Through biotechnology and metabolic engineering, scientists want to increase this amount, something Thelen aims to do. His end goal is to increase the oil in crops such as soybeans and canola, but any biotechnological idea for increasing seed oil starts in the model plant Arabidopsis. Modified plants that are being grown in Thelen's lab in Bond LSC. Photo by Mary Jane Rogers. Bond LSC. Arabidopsis is easy to transform in the lab and has a short life cycle, he said. We can use this plant to quickly demonstrate proof of principle for enhancing seed oil and then advance successful strategies to soybean, camelina, or canola. To increase seed oil content, Thelen's lab works on a large protein complex called acetyl-CoA carboxylase or ACase, an enzyme that catalyzes the first step towards oil production. We made a recent breakthrough on the regulation of this complex, Thelen said. The proteins critical to this process are called BADC proteins. They are akin to an essential part of ACase, but are inactive. BADC proteins significantly inhibit the activity of ACase by mimicking its functional sibling and slowing the complex down. Basically, BADC is a way for the plant to control and slow down the production of fatty acids. By turning off the BADC protein, ACase is deregulated and oil content in seeds significantly increases. We leveraged this discovery to make higher oil-producing plants by simply shutting down expression of this gene family by RNA interference, said Thelen. Consequently, this increased seed oil content quite a bit. We're now in the process of studying gene knockouts for this family in soybean and camelina. Yajin Yi, a postdoctoral researcher from China, adds water to the plants in Thelen's lab. Photo by Mary Jane Rogers. Bond LSC. Yajin Yi, a postdoctoral researcher from China in Thelen's lab, is in the thick of this work. He spends his time modifying seeds to maximize seed oil content and tracking the seed oil measurements in the GCMS instrument. The GCMS machine, which tracks the seed oil measurements. Photo by Mary Jane Rogers. Bond LSC. If you want to know how much oil is in each seed, you use this instrument, Yi said, pointing to the GCMS. It measures the oil content of the samples we provide it. Arabidopsis is a model plant used in Thelen's lab and the seeds are much smaller than most people would expect. Photo by Mary Jane Rogers. Bond LSC. Arabidopsis seed are much smaller than most people expect, so tiny and light that researchers have to be cautious that they don't become airborne and cause cross-contamination. In addition, soybean plants are kept upstairs in a fifth-floor greenhouse at Bond LSC, while camelina are grown in growth chambers within Schweitzer Hall as part of a collaboration with Dr. Abraham Koo, an assistant professor in the biochemistry department. Thelen's soybean plants, which are kept in the fifth-floor greenhouse of Bond LSC. Photo by Mary Jane Rogers. Bond LSC. Each of the soybean plants are transgenic and were screened for higher oil content as a result of BADC gene silencing. The plants are harvested every 3 or 4 months so the seed oil content can be monitored, said Yi. Thelen has begun patenting the BADC technology and another strategy for engineering ACAs to make it a more efficient enzyme complex. The BADC technology was co-invented with his previous graduate student, Matthew Saleh, who now works as a postdoctoral research associate at the Scripps Research Institute in San Diego. The patent examination process can take years, but if the technology is approved it would mean a huge influx of money in the agricultural market. The math is quite simple, Thelen said. A 1% increase in soybean seed oil translates to hundreds of millions of dollars. The numbers fluctuate depending on the market, but a 1% increase translates to about $200 million for soybean alone. A 5% increase, which again, I think is achievable, when realized across the diversity of oilseed crops, we're talking billions in added crop value annually. Yajin Yi, a postdoctoral researcher from China, with Vanildo Silveira and Caludet Santa Catarina, two visiting faculty from the State University of Rio de Janeiro in Brazil. Photo by Mary Jane Rogers. Bond LSC. His innovative approach has gotten the attention of scientists and researchers all around the world. The Vanildo Silveira and Claudette Santa Catarina, two visiting faculty from the State University of Rio de Janeiro in Brazil, came to Bond LSC specifically because of Thelen. Jay is an expert in the field and we wanted to work with him, said Silveira. At the moment, it seems like his research is on the right track. The initial data from Arabidopsis shows that silencing the entire BADC gene family substantially raises seed oil content, which is the main objective of his study. Preliminary, first-generation transgenics show soybean with higher oil, but these are greenhouse-grown. Randomized field trials will still be a while out, Thelen said. We're getting closer, but still a few years away from that goal. J.J. Thelen is a professor of biochemistry at MU and a researcher at Bond Life Science Center. He received degrees in both biological sciences and biochemistry, a B.S. from the University of Nebraska, Lincoln, and a Ph.D. from the University of Missouri, Columbia. He was a postdoctoral fellow at Michigan State University and has been at MU since 2002.